Hi, I'm Kate, and today I'm going to share a bit of my PhD research on the Greater Bilby and their social connections within a semi-wild sanctuary. The Greater Bilby is an Australian native fossorial marsupial that is active at night and digs burrows for shelter during the day. Their digging habits increase the productivity of nutrient-poor landscapes, making them an ecologically significant species in Australia. Bilbies are currently listed nationally as a threatened species and are thought to be solitary, but their behaviour is fairly difficult to study in the wild. It is more common that you will see tracks they leave behind than the animals themselves. I had the rare opportunity to study the behaviour of bilbies released to a semi-wild fence sanctuary in Dubbo, New South Wales. The fence excludes introduced predators that the bilby is particularly sensitive to, namely the fox and cat. The broader aim for this project was for the site to act as a source of semi-wild animals for reintroduction projects across Australia. Unlike more traditional breeding programs, animals were able to seek out mating partners and find their own food and resources within the fenced area. Bilbies were released in stages for the site, with the first release in October 2019, followed by a second release in December 2019. Bilbies were monitored post-release by tail mount attaching radio transmitters and GPS tags to bilbies to track and log their movements. DNA samples were also collected from founders and their young to assign parentage and estimate the breeding success of bilbies released. Prior to their release, bilbies were held in pre-release yards, and although bilbies are thought to be solitary, we observed some behaviours in the pre-release yards, such as this male scent marking, which indicate that bilbies are capable of leaving social cues for their conspecifics. This behaviour was first described in 1983 for a captive population of bilbies. Scent marking may be a key part of how bilbies navigate various social situations, but there has been no study as yet of bilby social behaviour outside of captivity. As a captive setting may intensify behaviour seen, this may not be indicative of bilby behaviour in more natural settings. Bilbies are known to share burrows, but we are yet to know the purpose of this behaviour and the potential benefits of sharing to bilbies. For solitary species, we predict that animals only socialise for the purposes of breeding, territoriality, or kinship, and that these interactions occur randomly. However, during the day, we found that bilbies shared burrows on 44% of days observed over 24 weeks of almost daily observation. And although the majority of observations were from mixed-sex groups, we also observed all female groups sharing and to a lesser degree all male sharing. This may indicate that burrow sharing may not solely be for the purposes of breeding in bilbies. A common strategy to boost the genetic diversity of reintroduced species is to introduce more genetics into the population. For bilbies, males need to seek out mating partners, and their breeding success relies on their access to females. If males from supplemental releases are unable to establish social connections with the resident females, which may occur due to competition with resident males, this strategy may not be as effective. My aim was to explore bilbies' social behaviour by using social networks to examine the strength of social connections between bilbies and release groups, and look at this relationship between male breeding success and sociality in a semi-wild context. Just like us, animals also have their own social network. Social networks capture the social interactions or associations that occur between individuals within a population. Networks contain a combination of dots and lines, also known as nodes and edges. Weighted edges are used to indicate the strength of social association between nodes, and this is generally depicted by the thickness of the line between two nodes. I constructed weighted bilby networks to plot the rate of burrow sharing between each bilby dyad or pair of nodes and the nightly proximity of bilbies. For nightly proximity, I looked at how many times two individuals were within 20 metres of each other, and where a GPS fix was taken for both individuals within a maximum of 2.5 minutes. I split the network into two time periods, before and after the second bilby group was released, to examine any changes in the network. By looking at the networks generated by the Bilby burrow sharing data, we can see variation in social association between Bilbies. Both before and after the second release, there seems to be a cluster of strongly associating Bilbies close to the centre of the network. Stronger associations, indicated by a thicker line, were also not restricted to male-female Bilby dyads, with strong association also occurring between female dyads. This may indicate that breeding may not be the only driver for social association in Bilbies. We can also see that all 16 Bilbies released to the site participated in the networks. The second release animals, circled in black, and particularly the second release males, were mainly on the outskirts of the network. If we look at the nightly proximity networks before the second release, all Bilbies seem to be associating fairly equally and the network looks fairly symmetrical. After the second release, the network appears to be more clustered, and unlike the burrow sharing network, second release animals were able to assimilate into the network, with some individuals located more centrally within the network. By comparing the mean weight of association between the sexes and release groups, we found that second release males had much weaker social association than all females and males from the first release group at burrows, but not in terms of their nightly proximity, with a more even spread of data for each group. This may indicate that Bilbies are more selective of burrow sharers, but not who they share space with at night, and that competition for access to females may be more fierce at burrow sites. By permuting the network 1,000 times and comparing this to the observed network indicated by the red line, I found that it was highly unlikely that the social associations in the observed Bilby network were occurring randomly. From parentage results, we found a connection between early breeding success and male sociality. All males that successfully bred had strong associations with females, and/or was more centrally located in networks. This means that male breeding success is likely to be connected to social position, and that stronger associations with females is likely to result in greater male breeding success in Bilbies. Despite our predictions, all Bilbies participated in the networks constructed. The networks indicate great complexity, particularly for a previously deemed solitary species. Social associations are unlikely to be occurring at random, and associations are not just restricted to male-female dyads. I plan to look at more potential drivers of social association in Bilbies, and we will be sequencing more DNA samples to improve our estimate of male breeding success and its link to male sociality within this semi-wild population. I would like to thank the following people and animals who supported this research, and I would like to thank you all for listening.